Hello, my name is Elizabeth. Thank you so much for coming to my poster. For some background, alcohol use disorder affects about 15 million people in the United States, and it's a progressive disorder that ranges from mild to severe. Some symptoms of alcohol use disorder include binge drinking or excessive alcohol intake and compulsive-like drinking, defined here as drinking despite negative consequences. The regions that we were interested in were the medial and lateral orbital frontal cortices. And we were interested in these regions because these are altered by alcohol use and are involved in alcohol drinking behaviors. And we can see them outlined here in blue and in purple. What we did to manipulate these regions was a chemogenetics approach, specifically DREADS. So we injected a viral vector into the MOFC or the LOFC. Then the HM4DI receptor was expressed on neurons, and only our designer drug, clozapine N-oxide or CNO, would bind to that modified receptor and therefore decrease neuronal firing. For our behavioral paradigm, the subjects underwent a drinking in the dark paradigm. Mice were given access to both water and 15% ethanol for a total of 12 drinking sessions first, as we can see in the gray boxes. And then where you see the arrows with the SAL, SAL, that's where they received a saline injection to habituate them to injections. So first they escalated their drinking on ethanol and water. Then on session 11, they were given a CNO injection to investigate the contributions of inhibiting these regions on baseline ethanol drinking. And then we tested aversion-resistant intake on sessions 13 through 16 in purple, where we added quinine, a bitter tastant, to the ethanol solution, and we gave a CNO or a vehicle injection in a crossover design on sessions 13 and 15. What we first found for the MOFC was that inhibiting this region did not alter ethanol intake or aversion-resistant intake. As we can see with the middle graph is that on session 10 and 12, these did not vary compared to session 11 when we inhibited the neurons in the MOFC. Then when we looked at aversion-resistant intake, we could see that there are no differences between groups when given a vehicle or CNO injection on ethanol and quinine consumption. In contrast, when we inhibited the LOFC, we found an increase in ethanol intake. So as we can see here, that compared to session 11, it is significantly increased compared to session 12, and it almost met the threshold of significance compared to session 10. And again, there were no differences observed when we injected CNO or vehicle on ethanol and quinine consumption. Another mechanism we were interested in investigating was the contributions of dopamine, because alcohol increases dopamine release in both the nucleus accumbens and other cortical regions. In addition, we've shown that D1 and D2 receptors are necessary for aversion-resistant ethanol intake. So what we did is we used a dopamine transporter knockout line. In addition, we also used a retrograde viral vector, which was injected into the nucleus accumbens core, and only neurons that express the dopamine transporter were infected, and these neurons go back to the VTA. What we first found was that for the DAT knockout mice, that there were no differences. So as we can see here, for ethanol consumption, there is no differences between our wild type and het mice. In addition, when we looked at aversion-resistant intake, there was no differences between our wild types and hets here as well. Next, when we inhibited the VTA to nucleus accumbens core dopamine transporting expressing neurons, we found that this may increase aversion-resistant ethanol intake. As you can see in the gray bar for the CNO Cre-positive animals, that only when they were given the CNO injection did they increase their consumption of ethanol and quinine solution. And this also almost met the threshold of significance with a p equals 0.0504 value. And histology is still pending. So what we can take away from this is that inhibition of the LOFC increases ethanol intake, but not aversion-resistant intake. And then there were no differences for the MOFC experiment. And then inhibition of dopaminergic neurons from the VTA to nucleus accumbens may increase aversion-resistant ethanol drinking, but we didn't see any differences when we knocked out all dopamine transporters in the brain. And more research is definitely needed to understand the contributions of these different regions and dopamine to aversion-resistant ethanol intake. Now I'd like to thank, of course, my advisor, Dr. Anna Radke, and all the wonderful undergrads who participated in this research, as well as our funding sources, especially the NIH and NINDS specifically. Thank you.